I'm John Strum, and this is RealTalk MS. It's December 29th, and welcome to this holiday edition of RealTalk MS. Speaking for myself, I am more than ready to say goodbye to 2020 and ring in a new year that holds a promise of happiness and health for the entire world. So I'll take this opportunity to wish each of you a very happy new year. ECTRIMS is an acronym for the European Committee for Treatment and Research in MS, and their annual scientific congress is the largest MS research conference in the world. For me, the best part of the conference occurs at the very end, when I sit down with Dr. Bruce Bebo, the Executive Vice President of Research at the National MS Society, to get his perspective on the highlights of the entire conference. And you've told me that this is one of your favorite conversations by downloading and listening to this episode of RealTalk MS more than any other. Well, this year, like almost everything else, ECTRIMS was virtual, and it was retitled as MSVirtual2020. But one thing about the conference was consistent with past years. For the third year in a row, you made Dr. Bebo's overview of the conference highlights your favorite podcast episode of the year. So in a moment, we'll revisit my conversation after the conclusion of MS Virtual 2020 with Dr. Bruce Bebo. Welcome back to the podcast, Dr. Bebo.
Thanks, John. As always, it's good to be with you. I look forward to this time after the meeting as well. So glad I could be here.
Let's get right into it. Which presentations or announcements at MS Virtual 2020 did you find particularly interesting?
Sure. So, I think the big story really here, John, is that MS research progress hasn't stopped because of COVID. There's advances in the development of new insights into MS, progress in the development of new therapies, learnings from the application of the current existing therapies. All of these things are helping to improve outcomes for people living with all forms of MS. So that's the overall story, John. We're still making progress despite all the roadblocks and hurdles that COVID has put up in our way. So you might remember from previous conversations we've had, and I think I've had on your podcast before, John, that the Society's developed a plan for advancing research towards a cure for MS, and that we calling this plan Pathways to Cures. And the plan really, it describes gaps in our knowledge, milestones, and accelerators that'll speed solutions and eventually a cure for everyone living with MS today, and as well as prevent MS for future generations. And this plan describes 3 cure pathways, so stop, restore, and end. And so during the meeting, there were advances reported for each of those pathways. So let's dive in, and I'm going to start with the stop pathway. So the goal of the stop pathway is to halt any new disease activity. So another way of saying this is no worsening of daily living or quality of life, and no changes in disease manifestations for everyone living with MS. That's the goal. That's what we're going for. And we've seen an evolution of disease-modifying treatments that's getting us closer to this cure. And at the meeting, there's a new target being investigated that I think is going to lead us to the next generation of disease-modifying therapies. And this new target, it's got a funny name like they all do, Bruton tyrosine kinase. Now, you don't have to remember that the way people mostly refer to it is BTK. So BTK, it's a cell signaling molecule. It's expressed in cells that we know contribute to MS, including B cells. Now, very promising data from phase 2 clinical trials of 3 different BTK inhibitors were reported at the meeting. The interesting thing about BTK is, in addition to regulating B cells, it also is important in regulating a cell called a microglia cell. And we think that microglial cells are involved in the compartmentalized immune reaction, the compartmentalized inflammation that happens inside of the nervous system. particularly important in the progressive phase or progressive forms of MS. And there was evidence presented last week that not only do BTK inhibitors regulate B cell function, but they also regulate the function of these microglial cells. And they also, there's some evidence to show that they could help promote myelin repair. So there's a lot of enthusiasm that this target and this family of medications could help stop relapsing and progressive forms of MS and might also promote repair of tissue. And all of these are advancing now to phase 3 clinical trials. For both relapsing and progressive forms of MS, we should hear something in that, you know how it goes. It takes a few years to do these clinical trials, but we should hear something more definitive about this in a few years. The results of a clinical trial of another agent, a novel agent called masitinib, were shared for the first time at the meeting. Masitinib is an inhibitor of another cell signaling pathway. This pathway also regulates the function of those microglial cells. Again, these are present in the central nervous system. They're there to normally help the central nervous system, the brain and the spinal cord, to fight infections, to help clear up any damage that might have occurred in the brain and spinal cord. And they're activated in MS. Interestingly enough, they're activated in other neurodegenerative diseases like Alzheimer's disease and Parkinson's disease. And they're thought to be really important players in this neurodegeneration. And that's what we think leads to progressive disability. So this was tested in a clinical trial of 656 adults with primary progressive MS or non-active secondary progressive MS. And it was found to reduce the risk for disability progression by 37%. And they looked at other measures of disability progression. They were also reduced. Nothing in the safety of this medication that was particularly troubling. So this is pretty exciting news, exciting particularly for people living with progressive forms of MS. And I'll be keeping my eyes on this. I know you will too, John. I know your listeners will be keeping their ears and their eyes posted for more information about that. And then before I leave the stop category, I also wanted to mention, it's too numerous to mention them individually, but this is a meeting that's also an opportunity for researchers and clinicians, and frankly, some of the pharmaceutical companies to share new data about the currently used disease-modifying therapies that are mostly approaches for stopping MS. And I think really the headline here is that our understanding of how and when to use these medications and approaches to treating MS is constantly being refined. And this refinement is leading to improved outcomes for people living with MS. So I'll stop there for a second. John, that's sort of in a nutshell, the highlights of things happening in the stop category. And I'll move on to restore, but I don't know if you have any questions or comments for me before I move on.
Maybe one. I'm curious what the next steps would be for a drug like masitinib based on where they currently are in the clinical trial process?
Sure. So, they've proven that this medication is relatively safe. They've shown in a smaller 656, doesn't seem like a small study, but in the grand scheme of things, that's relatively small, that this looks like it could be effective and is relatively safe. The next step is really treating this, testing this in a much larger cohort or group of people living with MS. So that's, as you know, and I'm sure many of your listeners know, that's referred to as a phase 3 clinical trial. So that's a definitive trial that has to happen, that tests safety in a larger group of people and tests effectiveness in a larger group of people. That gives us the best sense of the safety and effectiveness if the treatment was ever marketed, that it would be safe and effective in the population, in the real world, if you will. So that's the next step. Nothing shared at this meeting. All the signs indicate that this is a project that's ripe for taking to phase 3, and we'll have to just keep our eyes and ears posted to the news to see what progress they make for that.
I just recently got a question from a listener that sort of relates to this very topic. Masitinib is a good example of a drug that actually the clinical trials so far have taken place across Europe, outside of the United States. does that make it different in terms of ultimately seeing FDA approval if they should get to that point?
No, it's a great question. It doesn't, in the eyes of the FDA, it doesn't really matter to them where the clinical trials take place. What really matters is that they're done with rigor, that they're done in a scientifically rigorous way. The companies that perform these trials have conversations with the regulators, both the FDA for the United States and the EMA for Europe, prior to initiating these phase 2 studies, so that they know they're doing them in a right way that the regulators will approve of. And they certainly, after phase 2, have conversations with regulators like the FDA to make sure that their phase 3 trial is organized in a way and planned in a way that it will meet the requirements of the regulators for approval. So these companies are making hundreds of millions of dollars of investments in, particularly in phase 3. They tend to not do them unless they've had those conversations with the regulators and know that they're going to meet their requirements. And it doesn't matter mostly where the patient population is. They like to see diversity, right? So they like to see different groups involved in different jurisdictions, but if they take place in Europe or other parts of the world, it doesn't really matter. Well, let's move on to Restore, John. So the second objective in our Pathways to Cure roadmap is to reverse symptoms and disability. And in order to get this done, we're going to need to figure out how we repair tissue damage, especially we're figuring out how we're going to repair myelin. And so there were several advances in myelin repair that were shared at the meeting. And one of them was communicated by Robin Franklin. Dr. Franklin's a professor at the University of Cambridge. He's a recent Brancic Prize winner and was a featured speaker and presented the results from his lab that really focused on myelin repair. Now, there are two sort of main approaches to repairing myelin. One called endogenous repair, or another way of saying that is accelerating the body's natural ability to repair lost tissue. And then the other way to go about it is exogenous, or from outside. And that would be transplanting cells that potentially could promote repair. Now, Dr. Franklin's lab is focused on the former, on promoting natural repair, endogenous repair. And so what he's shown is that the nervous system does have this ability to repair itself, but that ability to repair itself is reduced as a consequence of aging, which is bad news for all of us. The good news is that the effects of age appear to be reversible. So he has shown in rodents that calorie restriction can reverse the effects of aging and accelerate myelin repair. And then maybe like going on to say, what it is about calorie restriction that does that? So he's gone on to show that a drug called metformin, this is a drug that's commonly used to treat type 2 diabetes. He's shown that this drug, metformin, can mimic the effect of calorie restriction in rodents. And this is an idea that's now being tested in humans in a clinical trial. And Dr. Franklin also shared something really interesting, John, that I hadn't really thought of before, but that the central nervous system, as we get older, gets less stiff, okay, just like mechanically less stiff. And he suspects that this reduction in stiffness might affect the ability of cells to repair myelin. So he's identified a receptor in myelin, in the myelin-making oligodendrocytes, those are the cells that make myelin, that can detect this stiffness, and it's called piezo-1. And he showed that you could target this pathway, and you can manipulate that pathway pharmacologically to promote myelin repair. So that's a pretty interesting thing to think of. Gianvito Martino, who's at the San Raffaele Scientific Institute, Milan, he's focusing more on advancing cell-based approaches to repair. So this exogenous, or outside, bringing cells from the outside to the nervous system. So he's shown previously that transplanted neural stem cells can produce factors that reduce inflammation, factors that could promote myelin repair, and also slow down neurodegeneration in rodents. So we can do a lot of things in rodents, right? But what we want to show is that it can work in humans. So he's now translating those findings into its first of its kind human clinical trial. So the first patient was injected with stem cells in 2017, and now he has 12 patients that have been injected with each with 3 doses of these stem cells. Dr. Martino reported at the meeting that he's not observed any serious adverse events. That's really the primary goal of the first trial in humans, is to make sure it's safe. So that's the way the trial is designed, to just make sure that it's safe. But he's also reported that the transplanted cells seem to survive. He's looked at as long as 3 months. They're still there. And again, while the study is really designed to look at safety, he did see I'd say characterized as a pretty dramatic shift in inflammatory markers in the cerebral spinal fluid. And I think this is a good sign that this approach might have a therapeutic benefit. And John, before I go to the end category in our Pathways to Cures, I think it's important to share that this is a super active area of research. There are about a dozen ongoing clinical trials testing strategies to promote myelin repair. I'm really hopeful that one or more of these approaches are going to show some benefit in people with MS and hopefully be available in the next few years. So any questions, John, or reaction or comments to the progress in Restore that we saw in the meeting?
Well, I just learned something new, that the central nervous system has great similarity to my catcher's mitt when I was in Little League. The older it got, the less stiff it became. So
exactly,
that was new information. A point of clarification for our listeners. When you talked about Dr. Martino's work, now those transplanted cells, to be clear, they are not the patient's own cells, are they?
Yeah, so they're from donors, and those patients, just like the rodents before them, have to be treated with immune suppressant medication so that those cells aren't rejected. Now, the nervous system's thought to be somewhat protected from the immune system in regards to or like transplant rejection, but to err on the side of caution and safety, those participants in those trials are treated with transplant rejection drugs so that those cells don't get transplanted. That's correct. But there are, and Dr. Martino and others are looking at ways of coaxing your own cells from your own body to be stem cells, and then coaxing them again to be myelin repair promoting cells, and with the idea that you could transplant those into your body and wouldn't have to have that hurdle of rejection associated with getting cells from a different donor.
Well, thank you for that clarification. I think we're ready to move into END.
Sure. So the third objective of Pathways to Cure is to end MS, and our definition of end is preventing new cases of MS. And I know we talked about this before, John, this concept that MS might be a preventable disease. And this concept's really starting to catch on. So this was the focus, and one of the piece of evidence that it's catching on is the Plenary Lecture. The first lecture, the biggest lecture of the meeting, was given by Helen Tremlett. Dr. Tremlett's a professor at the University of British Columbia, and her talk was focused on the prodrome of MS. That's the time before MS-specific symptoms develop. And there's a growing appreciation that MS has a prodrome, and she was looking for ways to maybe be able to identify people who are at high risk for developing MS and perhaps treating the disease before any specific clinical signs become apparent. So she presented data that showed that the pattern of healthcare use in people that later go on to develop MS changes in the years prior to diagnosis. So some of the more common issues that people went to the doctor for were pain, sleep disorders, anemia, and fatigue. So I'll give you an example. The odds of having a sleep disorder in a group of people that went on to later develop MS were 161% higher than in a group of people that don't develop MS. And people complaining of pain in this prodrome period had 115% higher risk of developing MS than in the group that didn't report pain in that prodrome period. And so you might also remember the last time we talked about this, we were talking about this sort of interesting observation that this levels of this emerging biomarker called neurofilament light chain, that they were elevated during this prodromal phase of MS and could potentially be used to identify people at high risk for developing MS. And there were a lot of presentations. This is a hot area of research. So there's a lot of presentations that advance our understanding of neurofilament light chain. But I would also say there are other biomarkers that are now being studied and explored that are emerging. So one of these was shared by investigators at the MS Center at Tel Aviv University, found that a specific immune pathway signature in the blood of people appeared during the prodromal phase of MS. And then another group that reported a new biomarker called GFAP appears to maybe also be a biomarker for central nervous system damage that could potentially be another factor that might be elevated during the prodrome. So I think the idea that we could identify people at high risk for developing MS before symptoms become apparent and treating them and potentially preventing MS from happening in the first place is gaining a lot of attention, a lot of work in that area, and I'm really looking forward to our time next year, John, when we have this talk again to see how much progress we made. And I did, if we've got time, the meeting was for, I think it was 3 days. It was Friday, Saturday, Sunday, and then there was a week, and then another session that was focused on reporting new data about MS and COVID. And so I thought maybe your listeners might be interested to hear what's happening and our understanding about COVID and MS. Does that seem like something you'd like to know more about?
Absolutely.
So there are many global efforts trying to collect data on outcomes of COVID-19 and people with MS. And these observations are focused really on trying to inform healthcare providers, the medical community, how we can best manage MS treatment or COVID treatment in people with MS. So I'm proud to say the Society has partnered with the Canadian MS Society and the Consortium of MS Centers to establish a unified North American COVID data collection effort for MS. It's called COVI-MS. We're not marketers, so we didn't, I'm sure we could have come up with a better name than that, but it works. Now, this collaborative effort up to now, John, has collected over 1,000 cases of COVID in MS. And this makes it one of, if maybe not the largest single collection of COVID cases in MS in the world. Now, up to now, the data has shown that the same risk factors for poor outcomes for COVID, like comorbidities like cardiovascular disease or diabetes or obesity or advanced aging, those same things also contribute to poor outcomes in people living with MS. And there isn't any data up to now that suggests MS on its own and contributes to any poor outcomes. Our data was presented during the meeting, the MS virtual meeting, during this COVID session, that reported that even after you correct for different rates of comorbidities and risk factors, that Black people with MS tend to have more severe COVID disease than white people with MS. And this has also been observed in the general population, and we really need to invest more resources into understanding why. Now, our North American COVID data collection, the COVI-MS collection effort, has joined forces with a program called the MS Data Alliance. This is a program that was initiated by the Multiple Sclerosis International Federation. And the idea here is to combine data about COVID and MS from all the different, or as many different global data collection programs as possible. So, of course, combining forces greatly increases the number and the diversity of cases and will allow us to more quickly answer the questions about how people with MS are faring with COVID and answer the question of whether any particular disease-modifying therapy contributed to either better or maybe worse outcomes. So initial data from this COVID-19 global data sharing initiative indicated that people who are older and live with higher levels of disability are more likely to experience a more severe course of COVID. They found no evidence that being on a disease-modifying therapy increased the death from COVID, but they did report that treatment with anti-CD20 medications. So this is Ocrevus and Rituxan. They did report that treatment with those medications was linked to more severe cases of COVID-19. Now, it's important to note that this data doesn't necessarily indicate that treatment with those medications caused poor outcomes. It only indicates that there's a link between people that take those medications and poor outcomes. And I would also important to point out, that other groups have looked at this and they've not seen this increased risk of poor outcomes in people on these medications. So there's kind of data in both directions. It's not clear exactly what's going on. What is clear is we have more work to do. And I would just suggest that if any of your listeners are worried about it, how their MS or their medications might alter their experience with COVID, they really should talk to their doctor or their healthcare provider. So just to summarize, John, I think, again, despite the disruption that COVID has caused to the world and the disruption that's caused to the MS research workplace, that we're still making really strong progress towards breakthroughs to a cure for MS. And I think we're closer than we've ever been to the day that we can say that we ended MS.
Well, I can't tell you how much I'm looking forward to that. That will be the last episode of this podcast, and I am ready to
I hope you'll invite me on for that one.
Well, of course I will. Who else is going to explain it right, So I'm looking forward to that episode. It's always great getting your perspective on what's become our annual conference recap. I'm looking forward to doing this again next year. I'm holding out hope that when we do, we'll be face-to-face at ECTRIMS 2021 in Vienna, Austria.
Yep, you and me both, John. I hope we get past all of this and we can be in person and be able to report on even more progress.
That's going to wrap up this episode of RealTalk MS. RealTalk MS is powered by the National MS Society, and you can share this episode of the podcast by letting your friends or family members know that all they have to do is point their web browser at realtalkms.com/174. You'll find that link in today's show notes so you can easily copy and paste it right into an email or text. And if you haven't yet, please don't forget to visit the Apple App Store or the Google Play Store and download the free RealTalk MS app to your iOS or Android smartphone or device. It's the easiest way for you to connect to the podcast. The app will automatically download the latest episode of RealTalk MS for you. You'll be able to save your favorite episodes, and it's a great way for me to share bonus content with you throughout the year. So I hope you'll take a moment and download the app today. I'm John Strum. Thanks for listening. Stay safe and have a happy new year.